Survival demands constant calculation. Every second, the human brain evaluates the surrounding environment to assign a biological worth to the options in front of it. When evaluating material goods, like food or money, the brain relies on a specific biological network. The ventromedial prefrontal cortex, ventral striatum, amygdala, and orbitofrontal cortex work together to compute what neuroscientists call a decision value. Yet human behavior routinely violates material self-interest. We share resources with strangers, donate to charity, and accept financial penalties to punish unfair behavior. If the brain's valuation system is built to maximize caloric and financial returns, why would it authorize choices resulting in a net material loss? Resolving this requires determining how our biological hardware actually processes an abstract concept like fairness. We need to know if the brain calculates social principles using the exact same mathematics it uses to count money. Neuroscience splits into two competing models. In the extended common currency schema, all incoming data becomes a universal metric. Financial payouts and social approval translate into identical neural currency, weighed together on a single scale. Conversely, the social valuation-specific schema separates these processing pathways entirely. This hypothesis argues that human interaction is exceptionally complex, requiring distinct populations of neurons that evolved explicitly to process social data, entirely separate from the neurons that count material goods. To determine which architecture the brain actually uses, we must look past behavioral observations and examine the functional MRI and pharmacological data directly. Researchers test these schemas by tracking blood oxygen level dependent, or BOLD, signals in an fMRI scanner, comparing the brain's oxygen consumption during monetary payouts against its consumption during social feedback. When subjects win a financial reward, specific activation geometries appear in the ventral striatum and the ventromedial prefrontal cortex. When subjects instead receive positive social approval, the exact same spatial patterns illuminate. This shared activation extends into vicarious valuation, situations where a person values an outcome exclusively from the reference frame of someone else. Merely observing another person win a monetary reward triggers activation in the observer's own ventral striatum, identical to the pattern produced if they had won the money themselves. This line graph plots the amplitude of the BOLD signal in the ventral medial prefrontal cortex over time. We are tracking two distinct decision values here, one where the subject chooses a reward for themselves, and one where they choose a reward vicariously for someone else. The signal amplitude for the vicarious choice aligns precisely with the personal choice. The prefrontal cortex actively tracks and encodes these decision values identically, regardless of the beneficiary. This architecture also absorbs highly abstract normative principles, such as inequality aversion. When two subjects play an economic game, a direct financial payout to player 1 triggers a specific level of ventral striatum activity. If player 2 starts the game with significantly less capital, that exact same financial payout to player 1 elicits much lower activation. The reward response scales dynamically based on the social context of the distribution, prioritizing relative standing over absolute numerical gain. At the macroscopic level, the brain integrates complex normative social principles seamlessly into its standard financial calculation pathways. The neuroimaging data heavily favors the common currency schema. However, regional BOLD signal overlap presents a specific mechanical problem. Macroscopic blood flow to an area does not guarantee that the exact same individual neurons are firing. To test the limits of this overlap, researchers used the trust game, an experimental design that isolates how humans learn from active social interactions compared to pure mathematical probabilities. This chart compares the neural responses during the social trust game against a non-social lottery game. When a partner violates a subject's trust, we see this distinct spike in the BOLD signal over the amygdala in caudate, correlating with decreased trust in future rounds. We can disrupt this specific learning pathway chemically by administering the neuropeptide oxytocin via a nasal spray. When the oxytocin condition is applied, the amygdala spike tied to the trust violation flattens completely. The subjects stop updating their behavioral trust based on the betrayal. Importantly, Learning in the non-social lottery game remains entirely unaffected by the hormone. The subjects accurately update their mathematical risk assessments exactly as before. This specific pharmacological dissociation shows that oxytocin selectively isolates and blocks social learning. It implies the existence of neurochemical pathways dedicated purely to social values, directly supporting the competing social valuation-specific schema. We see similar anomalies when examining learning signals. When a personal reward exceeds expectations, the ventral striatum generates a classic reward prediction error, instructing the brain to update its value estimates. But observe what happens when a subject models another person's choices. The ventral striatum pulses for the standard reward prediction, but simultaneously, the dorsomedial and dorsolateral prefrontal cortices activate. These cortical nodes are generating distinct action prediction errors. They are calculating the divergence between what the observed person was expected to do and what they actually did. The striatum is not managing vicarious learning alone. Valuing social factors requires massive amounts of external context. The ventromedial prefrontal cortex cannot perform these calculations in a vacuum. The computation relies on functional connectivity. This network graph shows bright pathways routing directly from the temporal parietal junction, a region deeply involved in social cognition, into the valuation circuitry of the prefrontal cortex. During a task like charitable giving, the temporal parietal junction calculates abstract variables like the perceived deservingness of the charity. It packages that data and transmits it along these pathways, actively modulating the output of the valuation regions. The strength of this physical coupling increases and decreases dynamically, driven entirely by whether the current choice context is social or non-social. These observations suggest the reward network functions as an integrative hub, relying on specialized data streams from social cognitive regions to form a complete value representation. We have macroscopic overlap across regions, distinct neurochemical pathways, and isolated prediction errors. To reconcile this, we must recognize the limits of our imaging tools. Functional MRI suffers from a resolution gap. A single distinct 3mm voxel highlighted on a brain scan does not represent a single mechanism. It contains hundreds of thousands of individual neurons. It is mathematically possible for both the common currency and social-specific schemas to be true simultaneously, simply operating at different scales of magnification. To prove this, researchers use single-unit recordings from non-human primates, measuring the electrical output of individual cells. At this cellular level, distinct neurons processing social rewards sit directly adjacent to those processing non-social rewards, intermingled within the exact same brain region. The macroscopic blood flow measured by fMRI simply obscures the microscopic cellular specialization. Both theoretical schemas accurately describe the biological architecture at different levels of magnification. This allows us to construct a unified systems model, answering the paradox of how the brain values abstract fairness alongside physical commodities. Looking at this unified systems model, the process becomes clear. Specialized regions like the temporoparietal junction feed highly specific social data into a shared macro network, which then routes it to distinct microcircuitry for calculation. The brain utilizes an extended common currency to execute the final decision, but the integers plugging into that calculation are generated by highly specialized context-dependent social inputs. There is no uniquely isolated social brain detached from our core economic drives operating independently. Empathy, fairness, and social behavior function as rigorous, computed variables integrated directly into our fundamental biological survival circuitry.